Hi, everybody. I hope you can all hear me okay. Yeah, perfect. Yeah, I'm Franziska and I run the Kaffeeklatsch mit Wissenschaft, or Science Communication Cafe at the Museum for Naturkunde since August 2019. So we've been doing this almost for 2 years, and I believe we've been mostly doing it online. And I think it's really nice that we get to reach so many of you wherever you are in the world right now. And it's been super fun, and I get to choose speakers that I find super interesting and really great. And that's why Diana is here today. Thank you so much for making time on a Sunday.
Thank you for the invitation.
I have obviously talked to you before and looked into the work you're doing. So I'm very keen to talk to you today about your work and what you do. And I'm pretty sure that everybody who's watching right now is gonna love you as much as I do after your talk. And it's gonna be even more interested in the brain, because what is that? It's not more like weird and cool and interesting. It's the human brain. So I'm glad we're gonna learn a little bit more about it today from you. So thanks so much for being here.
No, thank you for organizing this. I find this a really simple but also important platform, and also way to talk about science in a very informal context. I think it's the best way to remove the seriousness of it.
Yeah, and you can drink coffee. I mean, that's one of the main reasons I love doing this.
I am a tea drinker myself, but yeah, I have my...
We don't discriminate. That's totally okay. I have a few questions because, you know, I'm a very nosy person and I need to ask lots of questions before we even get into your research. And I have a bunch that I'm always really interested in. So my first one, as always, is if you are the first scientist in your family. Do you have role models in your family or close to your family that, you know, inspired you, or are you like brand new first generation academic?
My mom has a background in psychology, but she only did a bachelor's. And my sister and me, is very funny. They call us the scientists. Within my family's, within my dad's family side of the family, they call us the scientists, because we both decided to pursue. She did plant pathology, so she studied viruses, and she recently moved now to New York to do a postdoc. Oh. And we pretty much parted at the same time, so it was an empty nest.
Oh no, you poor family.
All of the sudden, but my parents have been always very supportive and proud of us. But we are the first members of the family that did my sister had a direct PhD because she studied in the US. That's how things work over there. Mm-hmm. And I did a master's first, and then a PhD. So role models, I think my high school chemistry professor was really, was an influence on me, definitely. And some other, no, I think at the end, my mom, I mean, she has a double major in psychology and social work, and she decided to, she chose the social work side of her career, and she didn't touch psychology never again. I guess also TV was a big part of our influence. Big man.
I can understand. I'm also the first one in my family to be a scientist, and I grew up watching, you know, Jurassic Park, Land Before Time, all of that. But it's really important to have teachers. I think sometimes people forget about them, because I also had a biology teacher that was so kind and so supportive. And I always knew I could come and talk to her when I had questions. So that's really neat that you had that. And so you are not from here. You're from Mexico, right?
Yes.
So
proudly.
You did Mexico City is one of my favorite places in the world. So we have to talk more about that later.
The jungle. So, it is, yeah.
Did you start out as a neurobiologist? Because did you do your bachelor's and your master's in Mexico? Because you said you left at the same time, but what is your background? Did you already start out as a neurobiologist? You knew that's what you wanted to do?
And no, not always. I wanted to be, I think from my generation, everybody had a phase in which they want to be marine biologist, and I had one. Yeah, yeah, exactly. And then it switched to medicine. And yeah, I think there's a very funny key moment for me. Yeah, I'm gonna talk about it. It's a little bit, I mean, it's funny. Yeah, it's a child's mind thing. I remember when I was a teenager and I first got my period, and I thought it was the worst thing in the world. And since then I got very committed, and I told my mom, just want to find a cure for it. So, because I thought it was a disease. I thought it was a disease, or at least I was like, yeah, so at least we can only have it once a year. You know, I wanted to be practical.
It sounds very practical, and probably something that lots of people have thought about, to be honest.
Yeah, so that got me into medicine. That really got me into medicine. And then when the time came, I had to apply to universities in Mexico, and I applied to one of the biggest ones, public universities, because my parents couldn't afford private education. But public education in Mexico is really good. And at the end, I ended up going to one of the biggest universities in Latin America for chemistry, because it's pharmaceutical and biological chemistry. Because at some point I thought, oh, do I really want to invest all these years? I mean, medicine is quite time consuming, and it's a lot of commitment, and you never stop. It's yeah. So I don't know. I think I put that into consideration, and then I compared the curricula to the major that I actually chose at the end, that was pharmaceutical and biological chemistry, and they were very similar topics, although they were more compact. And I ended up studying that. And in my last year of bachelor's, I had to do my thesis. And I ended up in a lab that was working with cell therapy on Parkinson's disease in a mouse model. Yeah. So using neural stem cells, these cells that have the ability to differentiate into the brain cells, because we don't only have neurons in our brain. Using them to compensate the degeneration that is happening in Parkinson's. And wow, that was fascinating for me. That was my first insight into the brain. And at the end of it, I was like, wow, I really want to know more. I just had a grasp of it. So when I finished my bachelor's, back then there was not a program in Mexico in neuroscience. Now there is a master's and a postgraduate program. But back then there was none. So I started to look outside of Mexico, and I just literally went on Google and I just typed neuroscience programs, masters or PhDs. And this little city in Europe showed up quite often, the Netherlands, because they had a lot of programs in neuroscience. And I started applying to a lot of them. And at the end, I got into Amsterdam and also into Rotterdam. Yeah. But because again, I didn't have the means to finance my studies, I applied for a full stipend for the National Council for Science and Technology in Mexico, and I was awarded with one that sponsored my master's studies. So I had the luck and the amazing opportunity to have that covered. And yeah, I went on a plane and I. Two big suitcases, without knowing anything about the Netherlands, other than they were very small and they had a lot of windmills and they ate cheese.
Yeah.
Exactly, and they ate cheese. And yeah, I made my way up there. Very nice, very amazing experience. The Dutchies are very nice people, very welcoming. And once I finished my master's, I was like, okay, I'm not done. I really want to continue. And I started applying again to grad school within Europe. And I was invited to several programs, but at the end I chose Berlin because after 2 years of living in Amsterdam, even though it's a capital, it's a little town. You just cross around it with your bike in 1 hour, within 1 hour. And
if you don't get run over, those people are vicious. When I was in Amsterdam, it's like, oh my gosh, I need to really, obey all the traffic laws here because
oh yeah, and it's so coordinated. It's like a dance. Yeah. It's so coordinated. Like there's no need of talk. Everybody just knows their way around. And you can tell when people are not from the city, when they're not locals.
They were scared looking, like I probably was.
Yeah. My dad also had a very similar experience when they went to visit. the first thing that I told him, do not stand in the bike lanes. The first thing that he did after stepping out of the bus is standing in the middle of the bike lane, and he was almost run over by a bike.
Oh no.
Yeah.
I guess it's a quintessential Amsterdam experience you have to have. So.
Yeah, and one that really changed my life because it got me into cycling. I mean, before I was one of the few ones because in Mexico, I used to live in front of campus. So for me, it was very easy to take my bike, and within 5 minutes, I was already there. And I was a minority then from people that were taking bicycles. And then in the Netherlands, I was like, okay, well, I have done this, but no, not for the amount, like the distances that were there. So I remember my first time cycling back and forth to campus, and I ended up completely exhausted. It was like a 20K. Yeah, I live very far away from campus. Yeah.
Wow. See, I mean, Berlin, I feel like we were getting better at cycling or being okay with people cycling, but nothing compared to the Netherlands. But I'm glad you ended up here. And yeah, I wrote down you were like Amsterdam, Mexico. You were in the US for a short time. Is that true?
Yeah, I was in Stanford in summer of 2010. I also got a stipend to attend the International Honors Program from Stanford University, and I took 3 classes. It was my very first international experience because I met a lot of Germans, people from Turkey, yeah, people from Turkey, China. It was exactly that, international, and yeah, it was great living the college life, the sorority, fraternity kind of like
of course,
style, yeah. And something that I always watch on TV and series, and then I had the chance to experience it by myself. That was really cool as well.
And when you looked up PhD programs and you found the one in Berlin, were you like, oh, okay, I've heard of Berlin, or because you lived in Amsterdam, so you're probably more aware of where stuff is in Europe, right? But did you ever think you would end up here, or did you think Berlin sounds good, I want to definitely come here? Or how did you choose Berlin as your new place to study?
That's a very good question. I didn't know. I mean, if you asked me 10 years ago, I've been in Berlin almost for 8 years. Yeah. And no, I never thought about ending up here for so long. After my master's, I started applying to a lot of programs, graduate programs, and one of them was the Max Delbruck Center program in Berlin. And I was invited for an interview. But before that, I had already been here once for a weekend visiting a friend when I was living in Amsterdam. And I loved it. For me, it was the first time I was like, okay, this is like a proper city with a proper metro subway system. Yeah, street food was a thing, which I missed a lot because in the Netherlands, it's not such a thing. In the Netherlands, you know what they have? They have like vending machines that are open 24/7 that are called vivos. And you put a coin and then it opens and you can pick a croquette, like the typical Dutch snacks.
Like hot food?
Hot food, but they're like, yeah, exactly.
We're lacking.
Yeah.
Oh, cool. Okay, so what else did I want to? Oh yeah, I think we mentioned this a little bit, but when did you decide to work on the human brain? Because you said, first of all, you wanted to, cure periods, but then you worked on mice, and you obviously, I mean, I think most of us think the brain is cool, but was that something that came quite quickly in your studies, or did you think you wanted to really do brain stuff for your entire career? Was it like that, or did you go back to marine biology briefly after a master's, thinking,
no, marine biology was left in the past. I had the chance to take a class in uni that was called Introductions to Genomics, and then there I learned about neurodegenerative diseases, and yeah, I was very fascinated by that, or the complexity of it, and yeah, the reason and yeah, the symptoms, all the things related to that. So for my thesis, I searched for a particular laboratory that at least was working with brain or brain research. And I found this one in particular. And yeah, since then it was like, I think I think my longest relationship, besides like my parents and my sister, that I had with neuroscience. I mean, I was very committed to neuroscience for a long time of my career. I mean, that's the only thing that I've been doing. Yeah.
That's cool. So maybe you can dive in and you can show us a little bit about what you
Yeah,
work
sure.
on. Share your slides with us.
Yeah, definitely. So today I'm gonna talk to you about a little bit of what I did in my PhD. It was protein trafficking. I like to call it knowing where to go because, well, as I will explain later, it's not always easy. But I mean, I prepared, a little introduction. I mean, I already talked with you about this, but I just want to recapitulate.
We love pictures here. So.
Yeah, okay, perfect. So yeah, I did a bachelor's in pharmaceutical and biological chemistry in the National Autonomous University of Mexico. That is the biggest public university in Mexico, and it's the largest university of Latin America. And yeah, here I am in the middle, the shortest one.
You're so short, yeah.
Yeah, I'm very short. Yeah, so yeah, it's not really. When I came to the Netherlands, I just felt like a midget, but yeah, I'm very short. Yeah, and then after that, as I said, I started to look abroad. I wanted to continue my education. I was very interested in the brain. Back then, there were no programs in Mexico, and the US was never an option, even though we're very influenced by American culture or like North American culture. Because for applications to university, you need to pay a fee, and sometimes they're quite expensive. Mm-hmm. And then I saw Europe and I was like, everything is free. And I was like, okay, why should I bother, right? So yeah, I got into a university. I got a scholarship. So I packed my bags and I just moved to the Netherlands. And then there I was fascinated with the tulips, the cheese, and the clocks. From September of 2012 to August of 2014, I did my master's in biomedical sciences at the University of Amsterdam. And besides my studies, I went really cliche, like touristic cliche, as you can see, like taking a photo in the tulip fields that I really can recommend. It's just fantastic. At least once in a lifetime, you guys have to have, I mean, if you can afford it. Now, I think they even offer virtual tours from the park that is there, so that's also very nice. And yeah, this is like the most Dutch photo that you can see, a windmill with the clocks. I loved it. I grew up a lot there within 2 years, also living by myself. After 25 years of living in Mexico City with my parents and having my laundry done and everything was ready on the table, I just went back to like traveling to a different country with a different language, a very cold weather, and being an adult all of a sudden, opening a bank account. Yeah, so after that, I'm a person that likes to learn constantly, and I'm still doing that. And I was like, this is not over. So in 2015, in January of 2015, after being interviewed and accepted into the program, I started my PhD at the Charité Universitätsmedizin in Berlin. Although, as doctoral students, the Charité only provides medical degrees. So as a doctoral student, you have to enroll into either the Humboldt University of Berlin or the Freie Universität Berlin. And I chose Freie Universität. I just don't remember why, to be honest. And I did it with a neuroscience focus. So yeah, before that, I would like to tell you a little bit more about where I come from, Mexico, and some interesting facts that are they're actually interesting for what I'm going to talk about next. So this is my hometown. This is where I come from. It's in the middle of Mexico, a little state. This is an aerial shot from one of the main avenues, Reforma. It's a huge city. It's a huge city. Yeah, this is one of my favorite places in the whole city. Is the Frida Kahlo Museum that is located in a very nice neighborhood close to my parents' place. And yeah, I mean, of course, because it's such a big city, we always face traffic. So this is a photo taken in December of 2017, very close to Christmas time, so Christmas shopping. And I mean, I don't know if you can tell, but this is a very also misleading photo because this is in red, this is in green. We were not moving. And I was like that for like 3 hours. And at some point I had to go to the toilet. I mean, it's very common around Christmas time to experience that kind of traffic because, yeah, I mean, there's a lot of Christmas parties from firmas and companies. There's a lot of shopping around because, of course, it's a commercial, very commercial time of the year. And yeah, I just remember being so frustrated and thinking, why am I here? Yeah. So a little bit of stats of Mexico City in 2019, and I took 2019 because, to be honest, I don't like to count, like many of us, I think 2020, we maybe don't like to count it in our years. A very interesting year, but also there was not a lot of traffic. So I think the data from 2020 couldn't really represent what is actually happening in the city because we were forced to be at home worldwide, Mexico City included. But in 2019, it was ranked number 13 in the world rank with the cities with most traffic. Wait, let me see if I can move this because, yeah, so how is that translated into hours that you lose in your day? So yeah, that is approximately 185 hours. That it's equivalent to 8 days of your life stuck in traffic. That is too much. So Francisca, you like to read, right? Yeah. So I found these very cool stats. So how many pages of have you read In Search of Lost Time from Marcel Proust? Okay, so you have read it more than once. That's how bad it was. If you like to, for example, knit, if that's a hobby for you, I know that it's a hobby for my sister, you could have knitted 49 hats and night sweaters. To be honest, I don't know who are they taking as a reference for this, maybe a very skilled knitter. And also
I think my friend Elizabeth I used to live with, she would knit like no one I ever met. She would definitely be able to do.
Yeah, my sister is also, I think she's connected. Shout out to my sister. She's also a very big fan of knitting. That's her hobby. You could have listened 3,748 times Imagine by John Lennon, a very beautiful song. And you could have cooked 5,639 pancakes or 9,384 cookies. You have basically organized a bake sale. And well, I don't know if you're into sports, now the Euro Cup is happening. You have watched all this amount of different sports matches. 112 football matches, 98 basketball matches, 130 American football matches. That was for me impressive because it's such a long sport, right? There's a lot of times, timeouts, a lot of advertisement in between, and also baseball is very long. And finally, I don't know if you're a Game of Thrones fan. Are you?
Yes, I also read the books.
Okay, I personally haven't seen it yet. I didn't jump into the train of Game of Thrones yet. But with this amount of time wasted in traffic that we spend in traffic in Mexico City, people could have watched 173 episodes of this series, this very popular series. And finally, do you like puzzles?
I mean, I like the idea of puzzles, and I did do 2 puzzles in lockdown, but I wouldn't say, I'm a puzzle fan. Yeah, it's more if it's here, I'll do it.
I think a lot jump into that train of puzzles during lockdown. I also did it. But anyways, average Mexican citizen could have completed 39 jigsaw puzzles of 500 pieces. So what am I talking about? In comparison to Berlin, in 2019, Berlin ranked 94 in cities with most traffic. And when you put this in perspective, while in Mexico City, you lose 8 days of your time, 8 days, 3 hours of your time in traffic. In Berlin, you lose 5 days. So basically 3 days less. And why am I talking about this? Because in my PhD, I basically studied traffic. So I went from living in traffic, as you saw in my photo from Christmas of 2017. I went from living on traffic to studying traffic. So why are cells and cities what do cells and cities have in common? How are they related? This is the generic illustration of a cell, the one that we can potentially find in textbooks and a lot of other content. But it looks very artificial, right? And one of the things that I love about the microscope, that is one of the most ancient instruments, is that it's still very current and very used in biology. And with the advance of technology and imaging techniques, we have been able to see and to find out that this is how an actual cell looks like. In gray, we have depicted here actin. I'm going to talk about later what actin is. In red, we can see microtubules. And blue, the nucleus, where DNA is located. And these are all images from the lab of Christophe Leterrier that he allowed me to use them. He's an expert in super resolution microscopy and a guru. He takes very nice photos. This is another comparison that I like a lot. On the left side, you have city lights actually taken from the International Space Station, from the NASA. And then on the right side, you have, neurons with fluorescent probes. And I mean, they look quite similar. I like this comparison a lot. I find it, I actually love it for the purpose of this talk, because I think, it represented my point. And I'm not the first one using this comparison, right? That cells are very similar to cities, but how exactly is this happening? How do cities compare to a cell? There's 2 points in particular. One of them is compartmentalization. And to depict this, I couldn't think of a better example of Marie Kondo. I don't know if the audience is familiar with Marie Kondo, but a couple of years ago, she got very popular on Netflix because she had a show, but she also wrote a book that is called Life-Changing Magic of Tidying Up. And this is Marie Kondo. So basically, she just showed up to people's places and helped them declutter the closets. So this was an example of a before image and after image. So she basically showed people how to organize and compartmentalize their closets. So I like to use this example because I think that's exactly what happens within the cell. So for example, in the case of prokaryotic cells, everything is in the cytoplasm dispersed, not really organized. DNA is all over. But in the case of eukaryotic cells, like animal cells, everything is distributed in membrane-bound organelles. And it's basically like eukaryotic cells, like eukaryotic cells are taking or adapting the Marie Kondo lifestyle and arranging all their structures within membrane-bound organelles and very organized in comparison to prokaryotic cells.
And the left one, can you go back once? So the left one is animal cells, right? And the right one is that plant cells?
This is bacteria. Prokaryotic is bacteria mostly.
Okay.
So
sure everybody knows.
No. Good that you ask. No, it's basically bacteria, and they don't have membrane-bound organelles, and the DNA material in comparison to eukaryotic cells that is present within a nucleus here, all packed, in prokaryotic cells is all over the place.
So it's just floating around in there?
Yeah, exactly. And the other thing, well, cells and cities are very similar, is because in a city, everything has a defined function, right? We have a post office, we have a factory, we have a supermarket. So all these buildings that are within a city have a specific purpose. We go to these places for a specific reason. And that's exactly what happens within a cell. So I'm going to go back to the generic animal cell. And for example, the nucleus would be equivalent to what a city hall does. that is basically controls what it's made within the cell and when. Then we have the cell membrane that will be equivalent to the city wall or the border, and it's basically, delimiting the city, or in this case, the cell. We have a Golgi apparatus that is like the post office. It basically packages goods or proteins inside a box and sends them out for delivery. The mitochondria is like the power plant, right? It produces energy in the form of ATP. And this energy is going to be used for other cell organelles to produce and to deliver their functions. We have the rough endoplasmic reticulum that is like the factory. It's basically what proteins are manufactured for exporting out of the cell. The lysosome, that is like the waste plant that breaks down waste whenever there might be some problems with how proteins are packed. In this case, that the lysosome is the organelle that is in charge of, is basically the dumpster of the cell. The vacuole, that is this blob over here, it will be like the water tower. It stores water for the cell. And the cytoskeleton that has two main functions. It acts as scaffold, providing support to the cell, but also as a sort of highways or tracks that proteins can use to be transported. And I'm going to focus a little bit more about also the cytoskeleton because it was part of what I studied within my PhD. So as I said, the cytoskeleton provided two things, right? One is cell shape and structure, so sort of like scaffolds for a building. So we have our generic cell. It looks like this, and if we remove everything, this is what it will be left, the cytoskeleton. So for us, a skeleton, of course, we are similar with the term, because as humans, we have a skeleton. So that is composed of bones that keep structure and supports our body. And within the cell, those bones would be these three fibrillar polymer proteins that are called microtubules, actin, or intermediate filaments. The cytoskeleton is very important, for example, in the particular case for neurons, because neurons are very well known for how long they can extend, right? They're like branched, like trees. They have thousands of branches, and they can elongate from our brain to connect to our muscles, like motor neurons. So I like to show this diagram because in vitro or in culture, when scientists take some tissue and dissociate it, and then you can obtain cells, single cells, and incubate them and keep them alive. In the particular case of neurons, cortical neurons, they start being very tiny, very round with these lamellipodial protrusions. And then there's a second stage in which they start extending immature neurites, they're called. And then in stage 3, it's called the break-up symmetry, because one of these branches is going to become the axon, and the axon is the longest extension of a neuron. And then you have on stage 4 a very well-defined dendrite and an axon here. And finally, in the latest stages, in the maturation stage of the neuron, they develop these little tiny things here that are there. These structures are very important for communication between neurons, and they're called dendritic spines, and they're formed later on. And these all protrusions and extensions, they are driven by the cytoskeleton.
And that is all happening in like a petri dish or like in a laboratory? It's just like they take it and then they just do their thing and grow?
Yeah, exactly. You can see them like that. Mm-hmm. And that's what I observe. Unfortunately, because it happened so fast, and that was not exactly what I was studying. I was not studying the branching of this, of neurons in particular. I don't have defined photos, but I found this scheme very nice. And that's what you see. So at the beginning of your culture, they're very rounded, and then the next day, overnight, they already started to extend. You have, of course, to provide them with a solid attachment, because if they are not attached, they're going to float and they tend to aggregate. So they form this aggregate, and we don't want that because in that case, it's difficult for us to study other aspects or individual cells like you want to study later, and it was my case. So for that, you have to coat your plate with special proteins that are addition proteins, and then in that you kind of like provide a substrate for the cells to attach, and once they attach, they can start branching.
And so quickly. That is really, I had no idea that works so fast.
Yeah, for the dendritic spines, at least for cortical neurons, that this is where I'm going. Cortical neurons are from the cerebral cortex. The cerebral cortex is the most outside part of the brain. And during the development of this structure, this is what is happening. So these are called radioglial cells that are, they serve as tracks for them to migrate. When they migrate, they are not so branched because otherwise it wouldn't be possible for them to go through, right? So they adopt this bipolar conformation, these neurons, and they just basically extend this process, the leading process, and then they sort of climb, you know, like when in your sports class, they made you climb the rope, sort of like that. They extend their arms, in this case, this leading process, and then they just use that to track their cell bodies. And then finally—
That's amazing, because thinking back, I mean, I feel like they're probably much better than I was.
Oh yeah,
in class.
oh yeah.
You just re-traumatized me thinking about how I had to climb up. Like we had both like a pole and like a what is it called, a rope, and the rope was even worse, okay, because it kept moving.
Yeah.
And
yeah,
they just
yeah.
do that much better than I ever could.
Yeah, and finally, once they reach their final destination within the cortex, I mean, the cortex is a 6-layer structure that, I mean, I don't exemplify here because it's not so relevant for my talk, but once they reach their final conformation, for example, this neuron is located in layer 5, they start branching, because if they were branched in this point, they wouldn't be able to move up, right? And yeah, the ventricular zone kind of like corresponds to here, subventricular zone, intermediate zone, cortical plate, and so on. So yeah, that's what happens in vivo. This is an example of what happens in vivo on a mouse brain. And this is how it actually looks like inside a skeleton in a mature neuron. In blue, you see depicted microtubules, and in green is actin. And I find also this photo beautiful, again from Christophe. In his
it almost doesn't look real. It almost looks like it's like animated, or
yeah, right,
like an
artist. That is, I mean, his lab in particular work with super resolution microscopy, so you are able to really see at each fiber, and that is fantastic.
Wow.
Mm-hmm. And well, the second part of the cytoskeleton, the importance of the cytoskeleton, as I said before, is that it supports a track or highway for protein transport. So basically, we have highways inside our cells, right? So if we take a look at this is, I exemplify here, part of the cytoskeleton is a microtubule. And when we go closer, if we take a microscope, and I'm going to show you that later, this is what proteins use to transport. They have like cars, basically, that are called motor proteins. And proteins travel within vesicles. So they travel within this, they're compacted, and yeah, they have like a package, basically. These are the deliveries. So in reality, this is how it looks like. And it's been confirmed that they move like this. This is a particular kinesin, and this is the motor part of the protein. And it walks. It basically walks along microtubule segments.
I'm sorry, but that is the cutest thing I've ever seen. I know people, as humans, we do this a lot, where we give like inanimate objects, like, you know, a trait, or oh, looks like a person, or does like a thing, or look, you know, but it looks so adorable just carrying, like, it looks just like it has a really hard day, you know, carrying that huge package. Just tracking along. It's,
Yeah.
oh my God. It's so adorable looking.
Yeah, I agree. I like this GIF a lot. It's very popular. It's very popular on the field. So construction of these highways in cells is happening constantly. This is another video that I took from David Sharp's lab, in which weekends in human cells, and with the help of genetic engineering, they were able to label with a fluorescent protein the growing tip of a microtubule, because a microtubule has a stable tip, so a tip that is not growing, and then units are added. So if you, there's a particular protein of family of proteins that are called EBs, and they're different. And this in particular is EB3, and it basically labels the growing tip of a microtubule. So you are able to see these comets. What we're seeing is growing microtubules. So in this way, we're able to study and to observe the dynamics of microtubules, and it's happening constantly. So this I also always found fascinating. This is, for example, this will be where all the minus, it's called minus and plus. The minus is the part that is not growing, and the plus is the part in which more molecules are added. So this is elongated. And yeah, it's happening constantly in our cells. So one way to, because this is happening constantly in neurons in particular, and because I told you, neurons have to change constantly the conformations to bipolar, to, and then later on when they are added dendritic spines, this has to be regulated. So like in a city, that for example, the city hall or the government kind of like hires a contractor for big projects. The city, the cell, the neuron in particular, well, all cells, but I'm talking about neurons because that was my field of expertise. Neurons kind of have their own contractors. And this is controlled by a family that is called rogitipases. And yeah, what they do is basically regulate the timing of growing or stopping of the cytoskeleton. And among this family, there's 3 members that are mostly, are more recognized, or let's say the more popular. One is called ROA, RAG1, and the other one is called CDC42. And these particular rogitipases, as I said, they're called the family of these proteins that regulates the growing and the building of these tracks. They also have to be very tightly regulated, because otherwise, if something is not working according to plan, chaos happens, of course. So in biology, how it looks is that a rogitipase is active when it's bound to a GTP, and then it's inactive when it's bound to a GDP. So that's what I'm depicting here with green and red. And they cycle constantly. They cycle constantly between the active and the inactive side. And this cycling is as well regulated by a family of proteins. The activation is regulated by a family of proteins called GEPs. And the inactivation is regulated by a family of proteins called GAPs. My project in particular was based on GAP proteins. If we translate back this to our cell city model, it will be basically like a building employee in which in an inactive state is just chilling and having coffee. And then in an active state, it has like a shovel and is ready to work. And their regulated gaps would be like the personal manager saying, okay, you're in, regulating the shift and saying, it's your turn. And then there's another personal manager that controls the pause breaks for this building constructor. That's basically how it works. So to make it more simple. Okay, all clear from here?
I appreciate your analogy so I can understand. It's something when I had topics like that in my studies, because I also did biology, my master's. Man, it's not so easy, you know, when you come from geology like me, suddenly you have to know this. And I feel like if my teacher had explained it to me like that, where would I be today? I don't know. But I really like the I really like how you explain this. It makes it much easier for me to.
And I had a lot of fun. For me, it was also a lot of fun. My dad is an engineer. He's always talking about these things. And I finally found myself using his talks and examples into something related to what I do.
Awesome.
Yeah. So my project focused particularly, as I said, in one of the GAP members in neurons. So one of these negative regulators, right? One of these person that is saying, no, okay, you take a pause. It's not your time to work. That is called NOMA-GAP. And this is the protagonist of my PhD. So NOMA-GAP is a GAP protein that is specific for the brain. And well, here this is where the talk goes a little bit more biological, but this is basically, you have to look at these two bars that explain that is basically only present in hippocampus cortex. This is a sample of the whole brain. And yeah, during in vivo, its development is kind of like decreases. It starts being active after P stands for postnatal day. And then 1 week later, you can see less expression. This is all at the level of protein, by the way. This is protein. This is basically if we do, maybe it's going to sound not so nice, but it's like if we take tissue from the brain and we just put it in a blender, and then we use antibodies that, well, I think most of the audience know that antibodies are proteins that detect specific proteins. In this case, we use an antibody against NOMA-GAP, which other name that goes by is SNX26. And we detect how present it is, its expression. And then it continues decreasing in the 2nd, 4th, and 6th week. And this is another way of showing that is only present within the cortex. Here, this is all working mice, by the way. And this is E stands for embryonic stage. And within the mouse model, for example, we have embryonic stages from 0 to 20. The gestational cycle from a mouse is very short. So they're born quite after 20 days, you have a bunch of little mice there. So around day 14th or 18th, you can see that it's increasing its expression. So in my lab, before I arrived, they have already seen that NOMA-GAP is a key regulator of cortical development. How they studied this is using a knockout model. And I know that when we hear the word knockout model, this is what it comes to our mind. But it's not like that. But actually, a knockout model, a knockout mouse is a mouse whose is not expressing one particular protein. And they can be 1 or 2, that is called double knockout. You can have a mouse model not expressing 3 different proteins, and that's a triple knockout. But in our case, it's a single knockout mouse that is not expressing NOMA-GAP. And it's a way to analyzing, basically removing something from the system and seeing what is happening when you remove it. So that's basically what our lab did. They removed this protein from the mouse, again, using genetic engineering. And it's a very lengthy process that I'm not going to go into depth. But yeah, this is basically the model that we had, and it was available already when I arrived to the lab. And basically what they observe is when you remove NOMA-GAP expression in the mouse, there was a significant decrease of the brain volume. These are all scanned photos from mouse brain model. And in particular from the cortical volume. So as I said, the most external area of the brain. And this was also observed by a different method, by histochemistry. And this here, again, is very obvious that these cortices in particular were, in the absence of NOMA-GAP, they were shorter, or they were not so expanded as in the control animal. But why was this the case? So again, continuing studies within the lab showed that this was actually because these neurons, the neurons that were missing NOMA-GAP, they were not as branched as the control animals. So as I said, a regular neuron has a lot of branches, it extends a lot, particularly cortical neurons, they extend massively. They have super long, the dendrite over here that is extending towards the edge, that's called the apical dendrite. They extend so long, and yeah, in knockout cells, they had not so many branches. So, and a way to study this, and this is a reconstruction from one of my neurons, is using a method called shoal analysis. And how it basically works is that you start drawing concentric circles equally distant of increasing diameters, of course. And then you basically count manually, although there are now some ways to kind of like semi-automatize it. But when I did it manually. And this method allows us to kind of measure the complexity of this branching. How many branches does a neuron have by counting the number of intersections? So for example, here, 1, 2, 3, 4, 5, and so on. And then you go with all the circles. And what you end up is with a graph similar like this, in which you have number of intersections on the y-axis, and then you have the distance from the soma, so each circle. And then basically it goes like that because the further you go away from the soma, the less amount of intersections you're going to have. And what our lab observed was, in the case of the knockout animal, this number of intersections was decreased. And this is just a graph showing that decrease. So basically these cortices were being shorter, and the volume of the brain was basically generally decreased because these neurons were not so branched. That's what we observed. Another interesting thing that the lab observed was that, again, in the case of NOMA-GAP knockout cells, there was a significant decrease of one particular surface receptor that is called GLUR1, that is very important for, again, communication between neurons, and it's expressed in the neuron's membrane. So this is a control animal, and what is important to observe here is the green panel. This is just an actin, and this is the merge of the green and the red channel. But I want you to focus on the green panel here. This is control animal, and then here on knockout animal, we have a decrease in the intensity, right? And this is basically portrayed here in this graph. So there was a significant decrease of this intensity. So then the question here was, if this is happening because there's less GLUR1 expressed, or is it having problems to reach its final membrane destination? And to answer this question, what we did again was take tissue from both animals and do another Western blot. This is a test that I already showed you before, in which basically what it does is to measure the level of expression of a particular protein. So we compare total level of protein towards surface protein expression. And while total levels remain the same, surface expression was decreased. And I know that it looks only like a band that is less intense than other, but it's actually a very important result, because basically we're showing that the total expression of the protein is there, that the absence of NOMA-GAP is not affecting the level of expression of GLUR1, but how much protein, GLUR1 in this case, reaches the membrane, that is the problem. So that made us thought, I portray this here. This is a control cell. Problem with my transitions, but this is a control cell. This is a NOMA-GAP cell, knockout cell. So basically depicting that there's less branches and there's less surface receptors expression. Yeah, here. So in our control model, we have a lot of receptors in the surface, and in our knockout model, just exemplified by one receptor. So this is where I came in. And this is part of, I'm going to talk about now a little bit of my actual results during my PhD project. So one thing that is important for us to understand, or for the audience to understand, is that proteins within a cell, they execute their function basically by interacting with other proteins in systems. And that's how biologists and biochemists have been able to identify particular pathways that are crucial for example, cell death or protein recycling, etc. And one way to do this at a protein level is with a technique called mass spectrometry. It's basically identifying NOMA-GAP partners or friends, let's sort of say. So what it happened in reality is that we took the the cortex of mice that were P7, so postnatal day 7, quite young. And then we lysed that tissue. So then we obtain a protein mix. And then we come in with a particular antibody that is specific for NOMA-GAP. And then in this protein mix, for example, NOMA-GAP is represented by this purple blob. So then the antibodies are going to come and recognize it. So they kind of like surround it. And then we use Sephiroth beads that allow us to kind of like capture the antibodies. And then after elution, so basically some washes, we're able to keep only our protein of interest, removing others. And then we run this on a gel, and then you see these bands. And then in this gel, you kind of like cut your area of interest, because basically this gel tells you when you're working with a particular protein, you already know how big it is. So big proteins in this gel are kind of like stay always in the top, and smaller proteins that are less heavy, they tend to run faster or for longer distances here. And I already knew that NOMA-GAP is a quite large protein. And after that, this little piece of gel, literally we cut a piece of gel here, and then we send it to analysis. And this is basically, to put it back again into like a more easier context, is like hiring a private agent, or actually several, because this test is quite pricey. So it's like you hire an agency, and then you basically go into your protein of interest, in this case NOMA-GAP, and you do the classic social media check, right? You're looking for the number of friends. So I have here friends of NOMA-GAP. We have all done this. In this case, it's like as scientists, we hire a very expensive private agency to look for friends of NOMA-GAP or interacting partners. So once we did that, we were able to identify that NOMA-GAP interacted with a lot of proteins that were related to vesicle-mediated transport. So remember this image that I already showed you of how proteins move within these vesicles. Well, NOMA-GAP apparently interacts with a lot of them, but this system is more complex. So one of them in particular that showed up in our analysis was a protein called RAB11. That is basically like a chaperone. It just like binds to the complex. The whole all this in like if you group it, it's called a complex, a transport complex. So it basically is part of the complex. It binds it targets the transport vesicle, and so we decided it showed up in our list, and then we decided to analyze a little bit more further into detail. So how we did this was to analyze the dynamics of RAB11. So what I did in the lab was to prepare a culture of control and knockout cells, and then using again biological engineering and genetic engineering, I labeled RAB11 with a fluorescent probe. And later on, I was able to study how it actually moves live. So if we use, again, I want to go to like, I want to explain it in a more simple way, how to study vesicular trafficking. So as I told you, we wanted to study the dynamics of RAB11. So what we did was basically to just tag it with like, let's say like a neon light. Studying vesicular trafficking is like playing hide and seek in the dark. So you tag and cheating a little bit as well, right? Because hide and seek, you are not supposed to know where the other person is, or like your person of interest. But it's basically, if we put a tag here on RAB11, and then we turn off the lights, and in the microscope, this fluorescent tag is gonna show up. So it's the signal that we're gonna detect in the microscope. And we're able to, or I was able to study how RAB11 was moving. And RAB11 was also in neurons is very important for the particular transport of this receptor that was decreased in the neurons that I told you about. So that was also very important to say. And yeah, well, basically, this is actual videos of my work, how it looks like. I have a control neuron here and a knockout neuron here, and I was able to study dynamics of RAB11 in living cells. And what we notice, or what I noticed in these videos, is that, for example, in control cells, there was a big accumulation of these vesicles in the soma, and they were not really going out to the dendrites. While in control neurons, it was not the case. The transport seemed to be quite efficient, moving up and down, etc. So, I mean, you might ask, how do you actually measure this? Because, yeah, this is complex data, right? And you have a lot of particles at the same time. How do you actually study this? How do you put numbers into this? So what you do first is that you select a segment that you want to analyze. In this particular cases here, a frame in red. And you obtain a graph like this that is called a chemograph. And I'm going to explain you what it is in the next slide. But it's basically a graph that tells you the position of a particle within time. And we analyze this in particular software that is actually open resources. So it's kind of like everybody can use it without paying. That's really nice. And what I observed is that in knockout neurons, there was an increase in the percentage of immobile particles. So basically what I saw with eye was confirmed with numbers. And also there was an increase in the speed of retrograde transport. What is retrograde transport, you ask? So for example, here from the soma to the periphery, so outside, that's called anterograde transport. And if you measure the other way around, so from the outside to the cell soma, that is called retrograde transport. So basically meaning they're going back to the soma, to the cell body. So we observe a decrease in the retrograde speed of these vesicles in the case of knockout neurons. But how do we actually obtain these graphs? I found this really nice video from the lab of Eugenia Trushina's, in which, this is how a chemograph is formed. So this is an axon in particular. I studied dendrites, but this is how we obtain these graphs, and we are able to study movement dynamics. So you have first a vesicle moving, and then is putting like a stop, or putting it into a two-dimensional conformation. Yeah.
Do we know how quickly, is this video like 10 times sped up, or do we know how quickly this happens?
Well, these videos, for example, are, my videos are are image 1 frame per second for
one frame per second.
Yeah, well, this is 1 frame per second, but what I wanted to say is that I speed it up. So this is speeded up 10 times.
Okay.
Yeah, it's not so fast. Well, when you actually
still fast.
Yeah, it's still fast. Yeah, definitely.
Yeah. Oh wow. Okay.
Yeah. Yeah. So this is how we obtain the chemographs, and then what happens when transport goes wrong? We were also able to notice in our knockout cells, in our NOMAGAP knockout cells, when we took a closer look at the apical dendrites, that there was accumulation of some structures called multilamellar bodies that similarly look like this. But when I tried to, going back to the city metaphor, they're basically like trash bags accumulated within the neurons. The question that, of course, is open in this case is this a consequence or an effect? So a cause or a consequence? Yeah, that's what I wanted to say. Is this a cause or a consequence? We don't know. That's one thing that is still open, but we were not able to see this accumulation in control cells. So it could be either that the transport of these vesicles, these RAB11 vesicles, is blocked by these massive structures because they're really big. When you see them with electron microscopy, that is a microscope that allows you to zoom in even more than regular microscopes. And then we were able to observe this degradation. These are degradation structures. That's what I call them trash bags. Because when something is not when the cell recognizes something as trash, that's when the degradation system kicks in. And then you have a process that is called autophagy, in which cell disposed or waste is recognized and then is wrapped in these structures. So yeah, that's something that's still open. We don't know. And then what I wanted to finish with is in my project, I was able to recognize a double identity for NOMAGAP, because I started telling you by with the fact that it was like a personal manager negatively regulating this CDC42 shift. So saying, okay, no, it's not your time to work. Just go out for a coffee, and you will be back when the GEF protein tells you. But I was also able to identify that it works as a traffic controller. So that was really cool. And with that, I also wanted to finish saying that the next time that you find yourself stuck in traffic, just think about the fact that you're not alone, that all the cells in your body are going through that. This is a really nice video from Professor Drew Berry from Australia. And yeah, you can see here, this is the actual speed, and this is a magnification of 10 million times. And these are microtubule motors. They travel up and down the cytoskeleton. And just think that, yeah, I mean, in your case, it's just like you're going to be there for a while. It's going to pass. You're going to get home. And the worst thing that can happen is that you miss some hours. But in the case of your cells, if something goes wrong, it can lead to disease. So with that, I would like to finish. Thank you for listening. It was really nice to talk with you about this. And yeah, I'm open for questions.
Thank you for your talk. I loved all the videos, I must say. I think you noticed. I'm very excited because it's a really quite difficult topic now. If you like, as a non-expert, you know, you listen to this, I'm like, oh
yeah,
okay, proteins. When I talk to my mom, who's a chef, when I say protein, she thinks of like chicken, you know, like, so for her,
okay.
when I said, oh, proteins, you know, and she's like, oh, and I was like, no, you know, like in the body, and she's like, oh, okay. So it's not so easy. So these videos really help me to picture what's going on, especially the little highways. Those were my favorites.
Yeah, I love that these resources are available, and I also ask for permission and stuff, but I love that people take their time to really make this visual, because I think it's important for the general audience, for non-scientists, for the non-scientific community to first not feel excluded. But I think science shouldn't be intimidating. I think everybody is able to understand science. The thing so far is that maybe as scientists we have failed to do so. But with opportunities like this, with platforms like this, like yours, I think it goes easier.
Yeah, it also makes you rethink about your work, right? If you have to,
yeah, completely. I
understand it. So.
I had a lot of fun. I had a lot of fun trying to find metaphors, and yeah, it was very nice to do this.
We have a question. If you have questions on YouTube, you can also post them there, and then we'll see them. But we have one here. It said, how do we know that this process works the same in mice and humans? How did you find out the removed proteins have the same effect in both? Did you find out?
So how do we know that this process works the same in mice and humans? Well, proteins have their siblings. For example, there's a lot of animal models, and genes in particular that are expressed in mice may have different names in humans, but they have the same function. So in the case of RAB11, I think the name is the same, and they act similarly. And how do we know how this process works the same? By basically just putting it into culture, or sometimes even slices, whole brain slices, and just analyze dynamics. It could be like the way that I did it, or well, at the moment I can only imagine that. And how did I find out that the removed protein does the same in both? In animals and humans. Okay, I get the question now. Well, yeah, that's a very good question, Curtis, because all the data that was available in NOMAGAP since until the moment that I started, and also 3 years within my PhD, was only available in mice. And then later on, we were contacted by a group of geneticists from the UK that have found point mutations in humans. And these point mutations were supposed to have, basically, what a point mutation is that you change one particular, you know, the genetic code. The genetic code has basically 4 letters. So A, T, G, C, and the cell can read it that way. And an amino acid that is a unit of a protein is determined by 3 of these nucleic acids, right? Sorry, not nucleic acids. I'm missing the word right now in English. But basically, the point mutation, what it happened is you change one amino acid within the protein, but the protein is still there, it's still expressed. But apparently changing this amino acid that is a unit of a protein, like for example, in the cytoskeleton microtubules, as I said, they're polymers, so they're a group of units. The same with proteins. They're also polymers of amino acids, right? So what it happens, or what they showed, is that this point mutation was supposed to have a bigger effect than the actual deletion of the whole protein in the mouse. So that makes you wonder. There's not a lot of data in humans until now that is starting. So I cannot really fully answer your question, but it is really interesting to see that a point mutation in the protein that actually is just, you have the protein there, but something in its function might be changed. We don't know yet, has a bigger effect in humans than the whole removal of the protein in animals, in mice in this case. So that might say that they might not function the same way, or they have different regulators. That can also happen.
All right. And you said that it can, like, you know, when you talked about the things go wrong and stuff that can happen, and you talked about diseases. Are there certain ones that come to mind where you're like, oh, this is what's caused in effect by that?
Well, a lot of neurodegenerative diseases, the hallmark of some of them, or a lot of them, is the actual misfolding of the protein that is not properly packaged in its box. And then because it's not packaged, it cannot be fully recognized, so it aggregates within a particular point of the cell. But in the case of neurodegenerative diseases, they cannot attempt to aggregate in the axon, that it is a very long extension that I showed you. So some of the most important ones, like Parkinson's or Alzheimer's, depend on these aggregations. And they said that, I mean, one thing is how they are formed, but because they're also not removed, that they may also be due, or that protein trafficking is also playing a role there.
Oh, okay. Yeah, I think those are terms that some people might have heard before. Okay. All right. So I have a few questions, and well, one of the questions that actually the first one that I could think of was, how was it in the beginning to work with mice? Did you have, you know, were you a bit worried about working with animals, or, you know, now you can be the one who can catch mice, you know, if they bother someone in their house, but in the past, were you kind of like, oh, I have to work with mice, you know, not everybody is likes them. Were you someone that was always fine with touching them or working on them, or is it something that came with time?
Yeah, another good question. I think I was at a very early stage of my career, I was in contact with rodents, because I think, my whole career I have only worked with rodent models. So rats first in my bachelor's, and then mice. The rats are like way bigger, right? So that was, I have to admit that was very intimidating, to be the first experience that I had with animals, because the only way that you have to obtain tissue is to, of course, sacrifice them. So I think for me, although it's not nice to think, I think for me it was clear that I couldn't get attached, first of all. and always to think that we're doing it with purpose. One thing that I could say is that in Germany, all animal work is very well regulated. So you even need to provide how many animals per year you're going to use. And if you use more than that, you're fined. So as a scientist, it really makes you think, how do I need to organize my work ahead of time? And how many animals within a year am I going to use, and for which reasons? So it really makes you organize yourself properly and to know basically waste animals without any purpose. Because as I said, the gestational period of mice is very fast. So if you put them together in a cage, you normally put one male and two females, or up to three as well. And then you might have a chance that that particular male mouse is very active, and then gets the three females pregnant. You need to have a purpose for those animals. You have to really make use of them, and that really makes you think in advance, to not waste any life without any particular reason. Mm-hmm.
It's good to know also that it's quite regulated, because I don't know if lots of people know about that.
Well,
like
within Germany, every state has its particular animal control regulation, but in Berlin, it is very strict. And that's always that I always heard from my German colleagues. In Berlin, it's very strict. There's a lot of paperwork in between, and it doesn't stop when the year is over. You can be checked 3 years later and say, okay, I want to see these experiments or these animal numbers. And then if they don't match, you can get into problems. Yeah.
And also, I'm just thinking about if something goes wrong, or like there is maybe a sick mouse that passes on illness. I mean, your whole experiment can just collapse, no? Is that something that you have to deal with? Because I know of friends who work with live, not always live animals, but also, like it could be anything from like little sea fleas or, plants or something, and then they're like a fungus comes and destroys all of their plants or whatever, and then they really have a huge problem. Or when they work with cells, everything's just destroyed. And
it's quite tricky. Yeah, working with animals is always tricky, particularly working with development stages, like I did as well with developmental stages, as I said. I mean, because there's three main kinds of cells within the brain. You have your dendrites, you have your astrocytes, and you have your oligodendrocytes. And then in the mouse in particular, they come like in waves. And first come the neurons, and you have a peak of neurogenesis that is called the production of neurons in mouse around E13, so embryonic stage for 13. The mama mouse pregnant is 13 days pregnant, and that's the peak of neurogenesis in the little embryos. So that means that you have to also be aware of that. But also some experiments, for example, you kind of like perform surgeries in embryos and you wait until they're adults. So that can take up to 1.5 months. And then if something went wrong and you found out 1.5 months later, you have to set another experiment and wait another 1.5 months. So yeah, it could be tricky. It could be frustrating sometimes. It can be time consuming. But again, it's a matter of organizing parallel projects, so you're never wasting your time. So okay, I have this surgery, but I also going to do this at the same time. So
so you're not stuck.
Yeah, exactly, because it can be frustrating. Yeah.
My next question is, maybe it's difficult to phrase it, but what is it like to work on part of the brain? What, you're thinking with your brain, and you're thinking about the brain, working on the brain. You know what I mean? This is sometimes you just look at your work and you're like, I'm just thinking with my brain right now about what the brain does. It's very meta, no? Because I have people that are neurobiologists, and they're like, sometimes I just wake up and I'm like, oh my God, my brain is thinking about the brain. It's like kind of weird. Do you ever have that experience where you're like, the brain is weird?
I mean, as I said in my final slide, as a person that has been in traffic constantly, and after studying trafficking within neurons, I'm like, okay, Diana, you're complaining about nothing. Like, you have tons of cells within your body doing all this parallel, like simultaneously. But I think because I always work with molecular biology, so a very specific side of not consciousness, not learning, not, I didn't do behavioral as well. So this question of like, I'm thinking with my brain about the brain, I think I had it like once or twice, but it was never like a full moment, you know, like
I had this, I'm asking because I had this, I was doing field work once, also 2010, so it was a good year for you and me. And I was doing field work and I found my very first dinosaur bone, and I just sat there kind of in shock. I was like, oh my gosh, it's, I mean, I study this and I know it's real, but then you find it and you're like, this, like I'm the first person ever to see this. I just sat there kind of like cool. Shocked.
For a brief period of time, I also consider paleontology, yeah, so
yeah, that's also a phase that lots of people have. But yeah, I kind of just like, I just thought, oh my god, it like either died here or it was, you know, like that's like the last resting place. I don't know, it was like a surreal moment where I questioned my entire existence, you know, because it's so old. It's a different kind of like, oh my god.
I think I'm going through that later. It's very funny, I'm going through that phase later after I finished my PhD. I was reading a book about psychedelic research to treat depression and how actually psychedelics work in the brain. And then I thought, wow, this is really interesting. Like to really get deep into the actual mechanism that somehow psychedelics are activating in your brain. It's just that was one of those moments, definitely.
Yeah, I bet. We have a question from YouTube.
Mm-hmm.
And I think maybe someone that came a bit late because we briefly like already talked about it, but someone asked, do you have a license to sacrifice mice?
Well, as a group, as a Arbeitsgruppe called in Germany, or like a working group or like research group, you have to apply for a specific experiments. So for example, all these experiments in imaging, I had to apply for that particular protein that I wanted to label. So yeah, it has to be very specific. And also as an individual, so as Diana, when I came to Germany, before touching animals at all, I had to take an animal course and I had to get certified. And then you're asked to every year attend continuous training, at least, I don't remember the number of hours, but you need to fulfill certain hours. So you're basically always updated on the latest trends, the ethics, and all the other aspects behind animal work.
I was gonna say there's also like always like an ethics committee, right? That checks if what you do is necessary or who it benefits and all that.
Mm-hmm.
All right.
We have a question, huh?
You have another question, I think, by the same person. How do we remove the fat in brain tissue while extracting proteins from the brain?
Good question. As I said, basically, extracting proteins from the brain is taking tissue. You take the actual tissue, and you kind of like put it through several phases. You first need to, DNA is also something very tricky that you need to remove, because if you don't, it gets very sticky. DNA is very sticky. But particularly for proteins, for the brain, I mean, you sort of not remove it if it's there at all. You just cannot target particularly proteins. So when I was studying NOMAGAB in particular to, for example, analyze the level of expression or these interacting partners with the use of antibodies. In that case, you kind of like create your complex and then you capture those antibodies and then you have your protein of interest. Mm-hmm.
It's several things you have to do to actually get there. It's not so easy.
Exactly. The mechanical dissociation as well first, yeah, to get single cells. Mm-hmm.
All right. If you have any more questions, you can write it in the chat. If not, I have a few more of my own. And I know you're not currently like super in the lab, but how in your experience will the work have changed? Because you're working or your working group work with animals, but right now they're still, I mean, it's getting a little bit better, but there are like social distancing rules. Are you even like, how does that influence the work in the lab? Because, you know, I assume the lab is not huge.
Yeah, working with, I mean, as coming from a group and a whole institute that is working actively with animals and particularly rodents, it was, I think for me, when the pandemic hit, I was already in my final phase. So I had to mostly do data analysis and just going to the lab to retrieve my huge blocks of data, because these videos that I show you just take a lot of space in your hard drive. But I heard from colleagues that, yeah, it was particularly tricky for them, especially when you have to maintain a mouse line, because it's not only you have to pay for that. That's another thing. If you want to work with animals, you need to have the budget to maintain your colony. It's basically, yeah, it's called a colony. To maintain a colony of particular animals, so you have to, I think it was put on stop at some point for a month and a half, and then afterwards, with the proper precautions, I think people were going, they were scheduling a calendar to not kind of like overlap with each other. But I guess, yeah, I can imagine that it's complicated, particularly when you're working again with development. So for example, if you're mating animals and you know that you have to take tissue at a particular embryonic stage, if you cannot be there, then you miss that stage. And then, if your previous experiment you did in that stage, you cannot replicate it because it's one day later. Or that's the tricky thing about working with development.
Mm-hmm. There's not so much working from home, right? Because like you said, you have to actually go there and like look after the animals too, right? And I think there's a good question from someone. I said, what's the worst disaster you ever had in your lab? I think that's a very good segue into that question. What's the worst thing that ever happened?
Oh, wow. Wow, that's a good question. I guess, oh yeah, once I remember something on the microwave. I was preparing a gel, and because agarose in particular, that's like Jello, it solidifies at room temperature. You need to heat it on the microwave, and I forgot it, and it exploded.
Yeah. So it's like the gel that you talked about earlier. That's you use.
That's a different kind of gel. That's with proteins. I was doing DNA, and it's a different material for DNA.
Oops. Did you get in trouble, or was it kind of like, oh no, we've all done this?
No, I think it was. I mean, it was also in my undergrad stage, so it wasn't. It was
expected.
Sort of, yeah.
That's a good question. I love asking those kind of questions because I also have one. It's like, what is the worst part of your job? Like, what is something that you're like, oh, if this wasn't part of my life as a scientist, I would be much happier. Is there something that you don't like?
Well, as I said, with particularly working in Berlin with animals, all this paperwork that you need to fill, that's not pleasant. So it was always something that I was not looking forward to, definitely. And yeah, so for example, there was this period in which I think there was a miscommunication, and some animals were not properly used, so at the end we had to get rid of them. But it was a one-time occasion, and we learned our lesson. And yeah, always that. I think, I mean, I have a lot of friends that I always like, I don't want to hear about what you do, and I respect that. And I think we respect each other, because I mean, I cannot change their way of thinking. I just think that that we wouldn't have any other chance to find out all the things that we know now. I mean, all this amount of research that has been done before my times. And I mean, it has been done using mostly animal models. So without animal models, we wouldn't have been able to have this amazing huge library of information that we have. So I think that's the forte, you know, the benefit, the advantage of studying with animals. Although on the counterpart is like, okay, I see the point. I see what people argue about it, and you have to choose your battles. And I guess I rather science advance. And it's always difficult to scale it up, always difficult. So recently, for example, with genetic engineering and the CRISPR-Cas model, and how studies were happening in humans, and how they were getting in a lot of troubles regarding ethics. It's always difficult. With mice, not so much.
My question, maybe that's a bit more positive, is what is the best part of your work? Because I think the worst part is something that lots of people that I ask say is paperwork, for example.
Yeah. The best part of my work, definitely, I think I worked there for such a long time that I built a very good group of friends they stopped being colleagues and became friends, like a close family. I would say that was very nice. And also, like having those moments in which your experiment really worked, or you found something that was significant for your research. Definitely getting these, for example, these videos is also not difficult. It wasn't so easy since the beginning. It was not that I did it one time and then suddenly all these little particles showed up in the microscope. You have to find your balance in expression. Sometimes I expressed too much and the whole cell was fluorescent, so I couldn't see single particles. So finding those right twitches in between and being, okay, I found the exact way in which I can see these vesicles. That was also amazing when you work so long on these neurons, and then finally you sit down and then you start imaging them, and then you see them fluoresce in front of your eyes. That's really great. That's fantastic. Mm-hmm.
So kind of like the discovery moment, we're like, oh my God.
Yeah, the eureka moment, yeah.
Someone asked the question that I also had. It's like, what is your future plan? Do you plan to stay in Berlin, or do you think about going elsewhere? Do you have anything that you want to do next?
I'm very I mean, I had to say I took a break, a long break that was very needed after the marathon that was the PhD. And I'm now actively looking for a job. I would love to stay in Berlin. I love Berlin. It has been my home, and it feels like now like home. But I'm not close to Germany in general, because it has been such a welcoming country, and I've been living here for a long time. I know the language now. I know how to move around. Yeah, it feels now like home. And I'm actually looking for a job. I would like to. I mean, I'm very interested in what you do. Science communication is another thing that I'm really interested, because I like to tell stories, and I like to include everybody in the stories. We saw it with the COVID pandemic, right? When the RNA vaccination started happening, and a lot of people were confused, and not everybody knew what it was. When you hear about you getting injected genetic material, of course, it sounds super intimidating, but yeah, it's not so like that. So I think there's a little gap there that I think I would like to help, or be part of the efforts that are being done at the moment, like you are doing it with this platform, to fill that gap between the scientific community and the rest of society. Mm-hmm.
Yeah, no, I can definitely understand that. I'm glad that more and more scientists want to, be part of that and want to talk about their work. So there's a bigger interest and understanding in science. I think, I must plug the museum. They're really good at having, like providing that. So I think it's really cool, because Cafe Clash is not the only thing they do, of course. And to reach people via podcasts and Instagram lives and all that, because, that's where the people are, especially when we couldn't leave the house. Like, everybody's basically attached to their screens now. I know I am all the time. And I have a last question, and I always like to finish with that one, because I wish I had someone tell me this when I was younger. And what is advice you would give a young scientist or inspiring scientist or someone who has maybe children that show an interest in science or anything like that, the natural world, what is advice you would give someone like that to pursue this as a career? Is there something that you think is advice that is good?
Oh, what a great question, huh? To also, what a great question to close with. I would say, I mean, it's going to sound like a cliche, but I wish I could have said that to myself. I mean, would you ask me at the beginning of our talk if I had a role model? Not really. So I would, I mean, my parents were always very supportive, but there was no, or when I was younger, I couldn't really relate to anybody going towards that direction, right? So, yeah, I said it was going to sound like a cliche, but don't be afraid to explore. Don't be afraid to ask questions. I think that's the most important. It doesn't matter how older or wiser you might think the other person might be. Don't be afraid to ask questions, I think, and yeah, we should believe in ourselves. I mean, we all have our drawbacks, but we all know also our values, and I think we tend to minimize ourselves. So yeah, the sky is the limit. I am happy. I mean, if we had talked 10 years ago, I would have never imagined that I would be sitting here in the basement of my parents-in-law house in the middle of the countryside in Germany. And I guess also be perseverant. It's very important to be perseverant and do not give up easily. I would say that that would be my advice. That applies to a lot of fields, but specifically in scientific field, because sometimes it can be not so positive or encouraging, but don't give up. I mean, if you feel that you can change something, just keep trying it. That's my advice.
That is great advice. And I see that someone from your family is also watching.
Yeah.
So
my aunt.
That is so sweet. So we have someone from Mexico watching. That's what I like about having things like that online.
Oh yeah, you have a lot of viewers right now. You have a lot of viewers from Mexico, in YouTube,
Yeah.
mostly. Yeah. So hola familia, gracias.
I love that. Yeah, I also have some friends over there, and one time we had a scientist from Argentina who then also had viewers from Argentina, and I love that because for some it's not so easy to see their family members talk about their science, right?
And I particularly told my parents to see it because I was like, I'm going to talk about it in a way that I think you're going to be able to understand it, because I think for 6 years they never knew what I was.
Oh,
what I was
my
doing.
family. They're
Yeah.
like, oh.
Yeah, my sister is there. Yeah.
Oh, I love that. Thanks everybody for watching. If there are not any more questions, I think we're basically in the time that we usually wrap up. So I think
Great, that was a lot of fun. Thank you for inviting me.
Thank you so much again for joining us today. I really appreciate it. I know you have a very busy week, like many scientists and science communicators. You have a lot of stuff going on right now. And we also have a Kaffee Klatsch again next month, of course. Yeah, it's in the chat. It's going to be on the 1st of August, and it's going to be our 2-year anniversary edition, because I think,
oh wow,
I think 2 years ago, my first Kaffee Klatsch ever was also on the 1st of August. I don't know if that makes sense, but something like that. 2 years ago, it's going to be in German. So it's going to be a German one again. But I'm also very excited for that one. I'm always very happy to talk to scientists about their work, and to you, of course. And if you want to give us some feedback, there's a survey also posted in the chat. So if you want to answer that so we know how we can do better or how you found us, then we would appreciate it. And I hope to see you all next month. And thanks for Bonnie and the team, of course, for always being
thank you
helping
to
us. And thank you for attending, and thanks for being here. See you next time. Bye.
Bye.